This is Biochemistry Unfolded, from atoms to algorithms, a weekly podcast on the topic of computational biochemistry and biophysics. Hello and good afternoon. Welcome to Biochemistry Unfolded, from atoms to algorithms. And so today, in today's episode, I'm going to talk about a particular algorithm. It's also an artificial intelligence algorithm. And so this is the title of this paper is De Novo Designed Proteins Neutralize Lethal Snake Venom Toxins. The primary author is Susana Vasquez-Torres, and there's a bunch of other authors in this paper as well too. This also includes David Baker, who recently received the Nobel Prize in Chemistry last year for his work on Rosetta fold and protein engineering predictions. So this study presents a fairly novel approach to lethalizing snake venom toxins using de novo designed proteins. Traditionally, snakebite antivenoms are derived from immunized animal plasma, which has significant limitations. Some of these limitations include relatively high production costs. If you've ever been bitten by a snake and you've ever seen that you've ever seen the bill from that can be fairly high. There's also a fairly limited efficacy against certain toxins, such as three-finger toxins and 3-FTXs, which are talked about in this paper, as well as there's accessibility issues. In this study, what the authors do is they leverage a deep learning-based protein design to create highly stable, manufacturable protein binders that bind to that snake venom and neutralize it with very high specificity and efficacy. So this is fairly important. Their approach could very well lead to a safer, cost-effective alternative to traditional antivenoms. And so that's very important. I kind of, when I was reading this paper, I was kind of thinking of Samuel L. Jackson, who's also he's actually a native Tennessean here. He was in the famous movie Snakes on a Plane, where he was quoted, and I'll sanitize the quote a little bit, but I think he talked about, "Enough is enough. I have had it with these mother effing snakes on this mother effing plane." And then he shot the windows out and the snakes all flew out. Let's see if we can solve the snakebite problem without having to blow out the windows of an airplane. Now, if we look at this particular paper, I actually want to bring up the first page of the paper. This was published fairly recently. It was published online in January 2025. Was actually received late last year. If we look at the authors on this paper, we can see several authors, and you can see their institutions and where they come from. I think it's important to give credit where credit is due, that it's not just one person doing the science, it's many people. And then what I also want to do is look at the very end of the paper, the last page. I think I went a little bit too far. Okay, yeah, so under acknowledgments. And this is also important because this is where the funding for this study went through. And if we look at the funding for this, you kind of realize where that there are multiple sources of funding. First of all, of course, David Baker's lab in Washington State, they depended on the Washington State General Operating Fund, which is state tax dollars in that state, for supporting David Baker's Institute for Protein Design. There's also a couple of nonprofits involved, such as the Audacious Project and the Howard Hughes Medical Institute and the Novo Nordisk Foundation as well. There's several European sources of support, both government and as well as foundations in the United Kingdom. Also here in the United States, we have the National Science Foundation, so they supported this research as well. It's also supported by the NIH, National Institute of General Medical Sciences, and the Department of Energy also funded this study as well. So there is several support both here as well as in Europe. It's very important to acknowledge this research because a lot of this science supports the economic activities of the entire nation. And so whenever we spend, information, I think Francis Collins recently cited that for every dollar of funding that's spent to the NIH from the NIH, that delivers $141 of economic output from those studies. So that's very important to point out. I think since World War II, I think 50% of the economic output of the United States can be traced in some way, shape, or form to our investment in science. So I think that's very important to point out the economic output for many of these papers that we're doing. And I think when we look at snake venom, this will certainly have an impact because, snake bite, the snake bite envenoming is a fairly big neglected tropical disease, but it doesn't only affect the tropics. There's also a lot of snakes in the United States as well. And we're certainly not tropical, although. So overall, snake bite can result in over 100,000 deaths annually. So that's some of the statistics that we're facing here. In particular, this paper goes over 3 finger toxins, which they abbreviate as 3FTXs. And see, these are highly topic venom components that cause neurotoxicity and tissue damage. Currently, the treatments rely on animal-derived polyclonal antibodies. These tend to be fairly expensive, so that's where most of the cost is. They're also ineffective against specifically the 3FTX excess neurotoxins. So what the authors did was they proposed de novo protein design as a solution for creating high affinity toxin neutralizing proteins. And so the advantages here, of course, we don't have to rely on animal immunization methods, and we can produce these proteins via recombinant DNA technology. And the other main advantage is we have their small size, which enhances tissue penetration, and there's a very high thermal stability as well, and they're fairly inexpensive to produce because, again, recombinant DNA technology, we can produce that fairly inexpensively. Of note in this particular study, they used an artificial intelligence or deep learning based method called RFDiffusion. That came out of the David Baker lab. And so what they used RFDiffusion for was to generate binders targeting alpha neurotoxins as well as cytotoxins. So if we look at those targeted alpha neurotoxins, these inhibit the nicotinic acetylcholine receptors, and they cause those are in the muscle, and they essentially cause paralysis. So essentially, the snake is using the venom to paralyze its victim, and so then it can either use that for food, consume whatever it just paralyzed, or they're using it for defense to keep away an animal that's more dangerous to keep it out. So this is a computational design method, and so they created the proteins that bind to these toxins, and then that block their interaction with the receptors. They selected 44 candidate proteins in the paper through a yeast surface display screening. And then the top candidates were refined through optimization. And they achieved a fairly high binding affinity. The dissociation constant for the best binder was about 0.9 nanomolar. That's the SHRT binding protein. Nanomolar binding affinity, of course, the lower the number, the smaller that number, the tighter binding. And you want, in this case, the protein we want to use a fairly tight binder so that it can neutralize the antivenom protein and take that out so it doesn't cause cell damage. That's the whole goal ultimately. And so the cytotoxins, these cytotoxins that are in the Venom, they destabilize the cell membranes, and that leads to severe tissue necrosis. The word necrosis, of course, means death, right? So cell death. And so the design proteins will bind directly to critical toxin loops that prevents membrane insertion. So we generated the authors of the study generated 55 protein designs. The best candidate neutralized venom from multiple cobra species. So these venoms are part of that cobra. That's the snake with the hood, right? Now, they didn't just do, in this paper, they did not just do computational studies. They took these proteins that were being designed computationally and then went ahead and actually synthesized them for in vitro and in vivo experiments. The in vitro experiments involved SHRT completely neutralizing alpha neurotoxins in cell studies. This was in human-derived muscle cells. And the CYTX protected about 70 to 90% of human keratinocytes from venom-induced cytotoxicity. The in vivo experiments are very interesting because mice pre-treated with the binders survived lethal venom doses. So that was very important. We want to see very high survival rates. And even mice treated 15 to 30 minutes post-exposure also had very high survival rates. Because again, many times you get bitten by a snake that you may not have that antivenom right away. So 15 to 30 minutes later, that's actually a pretty good amount of time. So that's good. So these design proteins, they show comparable or even in many cases superior efficacy to monoclonal antibodies. And so the key advantages here, we have scalable production without relying on animal-derived plasma. We have a very high stability, and that allows storage without refrigeration. And again, the cost effectiveness of using these proteins versus animal immunology studies or animal antibodies, this is going to lead to a much lower cost. And that's going to benefit not only those of us in the United States, our insurance companies will not have to spend as much money, but also this should impact those in the third world where the financial resources are not quite as available as well. So there can be future research in these studies as well that might extend the RFDiffusion-based design to other medical applications. This could include neutralizing bacterial and viral toxins. So we could have novel antibiotics designed this way, or even antiviral drugs, right? So how does RFDiffusion work? Let's talk a little bit about that software. And so this was produced out of the David Baker lab. And so RFDiffusion is an artificial intelligence method. It's based on deep learning for generating new protein structures from scratch. Literally from scratch is what de novo literally means. And so we're iteratively refining a randomized starting structure into a functional, well-folded protein. And so what it is, it's based on diffusion models in artificial intelligence. And what is a diffusion model? If you're familiar with image generation, such as using DALL-E, or maybe if you're using like Adobe Photoshop, when Adobe Photoshop's AI can generate an image, we're adapting these same techniques that we use for image generation to generating novel proteins. So how does this diffusion process work? So the diffusion models work by starting with random noise, and they gradually refine that all the way down into meaningful structures. So in RFDiffusion, step number one is that the algorithm starts with a random set of amino acid residues that have no structure. Step two, we're going to apply reverse diffusion steps to gradually transform that structure into a folded protein. And in step three, that model ensures that the generated protein has stable interactions as well as a biologically relevant shape to enable binding to the protein target of interest. And it does this by conditioning it on known protein folding rules, right? Now, what makes RFDiffusion unique? Number one is its secondary structure control, where users can specify the desired structural features, such as alpha helices and beta sheets. Number two is that we can have specific targeting of the binding site. This can be guided to generate proteins that bind to specific targets, such as venom toxins. It also uses beta strand pairing, which can generate proteins that form very strong interactions with the target molecules. And then we have an iterative refinement that ensures that the final protein is well folded and stable. So how did they do this in this particular study? In the snake venom study, they started with input data. The researchers provided structural data for 3-FTX venom toxins. And in the design phase, the RFDiffusion generated protein binders that complemented the shape of these toxins. We then moved on to a filtering and optimization step. So the best candidates were then refined using AlphaFold 2 as well as Rosetta, and that ensured stability and strong binding. And finally was the experimental validation, and that's where the top designs were produced and tested, showing high affinity binding and venom neutralization. So what this does is it revolutionizes protein design. There's no need for trial and error in lab experiments. It reduces the costs, so the AI-driven design avoids expensive screening of millions of molecules, and we can open up brand new therapeutic possibilities. We can now create custom proteins for diseases, toxins, antibacterials, antivenoms, and so much more. So that's basically the gist of RFDiffusion. And so we have a relatively big breakthrough in computational protein design, and that has implications far beyond snakebite treatment. This paper demonstrates the power of AI-driven protein engineering to create therapeutic molecules. We revolutionize antivenom development, and that offers a more effective, affordable, and scalable solution. We also have potential applications in other neglected tropical diseases, and toxin neutralization strategies. So one thing, if we kind of look at this paper in a little bit more detail, figure 1 of this paper kind of shows the antivenom. And so figure 1A shows the structure of 3-FTXS neurotoxin, and that you could actually download from the protein data bank. That's structure 1QKD. And so in that structure, there's some very highly conserved cysteine residues. Those are kind of shown in yellow there. Those are the little linkages between proteins. And so those are kind of linking the different beta sheets in there. Up under B, that shows the cytotoxin, that same cytotoxin, how it actually breaks apart the cell membrane. And so that leads directly to cell death, because if the cell membrane ruptures, then the whole cell components leak out, and then we have a bigger problem right there. Under C, now what's happening there? That is the nicotinic acetylcholine receptor in muscle cells. And so the proteins are, you can actually see the acetylcholine binding site in that nicotinic acetylcholine receptor. And so the protein kind of, the design proteins are designed to bind on top of that to prevent blockage of this channel right here, right? And so under D, that just kind of shows how the designed protein in gray is actually going in, and it kind of blocks the antivenom, which is, the antivenom's under A. So the antivenom is in purple under D there, and the design protein comes in and it blocks, it binds to that antivenom, so it has a different shape so that it cannot bind to the nicotinic acetylcholine receptor, right? The experimental data is very interesting, and that's shown under figure 2. And so what's interesting here, they used 2 specific techniques. Surface plasmon resonance. So let's just kind of look here, surface plasmon resonance. And so what is surface plasmon resonance, right? This is an optical technique that measures how molecules interact. And so it's very commonly used to study biomolecular interactions, such as protein-protein interactions, as well as ligand binding. So how does this work? First of all, light hits a metal surface at a very specific angle, and that light excites electrons in the metal, causing them to move parallel to the surface. Then those moving electrons create an electric field, and the electric field, the range of that is about 300 nanometers from the metal surface. So the reflected light changes when there's a change in the refractive index of the material near that metal surface, right? And so if you kind of look at the surface plasmon resonance studies under C, so we've got the different, we have SHRT in the first row, LNG is the second row, and CYTX is in the third row. So these are all the designed proteins. And then in the column C under SPR, that is what the SPR data actually looks like in this case. And you can also determine the KD value. Again, the lowest KD values indicate the tightest binding between the designed protein and its neurotoxin. So 0.9 nanomolar is a very tight binding. Even 1.9 is fairly tight binding as well. 271, that's like 0.271 micromolar binding, so it's not quite as strong, but it is still a fairly tight binding. So maybe more research could be done on CYTX to see if that can be improved as well, right? So that's how surface plasmon resonance works. The other technique that they used is called circular dichroism, right? So, what is circular dichroism? Circular dichroism is a method that measures how light is absorbed by a molecule to determine its structure. And so, in this type of spectroscopy, we're studying proteins and other biological molecules, similar to SPR. How does this work? Well, CD measures right and left-handed polarized light. That's what we call it, circular dichroism. So it's kind of like a circular polarimeter in a sense. And so these, those right and left-handed polarized light is absorbed differently by the molecule. The transmitted light becomes elliptically polarized, and we have a difference in absorption is measured by its ellipticity, right? So what we can do with circular dichroism, we can actually determine the secondary and tertiary structure of proteins. We can confirm whether an alpha helix or a beta sheet exists in the protein. We can also study how proteins fold, as well as how they're being denatured. And we can also determine how mutations in ligands might also affect the protein stability and how metals and other molecules interact in the protein as well. So currently, CD-SPEC is used in drug research to analyze antibodies. We can determine isoelectric points and a lot more information. We can use this to study the structures of polypeptides. And in this case, we're confirming the structure of these designed proteins as antivenoms, right? So the circular dichroism data, we can kind of see that in the far column under D, and kind of see the wavelength there with the different levels. And so that's what the circular dichroism shows us. And in this case, they actually found, we've confirmed the presence of an alpha-beta secondary structure in the 3-FTX binding protein, and there's thermal stability. The theta value is the molar ellipticity. So we're plotting the molar ellipticity in those graphs as a function of wavelength, right? So the experimental data, again, that confirms the efficacy, and it confirms that these proteins actually do what they're supposed to be designed to do, which is particularly notable. I found figure 3 very interesting too, because they actually crystallized these complexes. And so they grew X-ray crystal structures, determined that data, and they found that for these 3 design proteins, the X-ray crystal structures actually matched the predicted structures from RFDiffusion. And so the proteins not only inhibited the neurotoxins from the snake venom, they also did it in such a way that matched the design protein effect, the design protein structure. And so that was also very good. That kind of gave a little bit of a validation to the RFDiffusion method in determining, in finding these proteins' structure, right? And so the in vivo studies, that's under figure 4 in this paper. And again, they found that for the most part, those mice did have a high rate of survival when they were injected with various neurotoxins. And so that was also particularly valuable as well. So I think we've got a very interesting paper here. And I think this shows the effectiveness of using AI-driven protein engineering to create relevant molecules, in this case to treat snake bite and antivenom. And it kind of offers a more effective, affordable, and scalable solution. And this should have a huge effect moving forward And we can even use these techniques to help treat other diseases where other proteins come into play, right? So Alzheimer's, Parkinson's, antibacterials. There's a whole host of things that we could use these proteins for. And so I hope you found this paper very interesting. I hope you found this episode very interesting. And if you kind of like all these videos, please leave me a comment down below. If there's a topic you'd like to learn more of, let me know about that as well. And go ahead and hit the thumbs up button if you like this video. Also, please subscribe to this channel and hit the little bell icon on there if you'd like to be notified of upcoming content. And so for now, I will let you go, and I will see you next week in the next video. Thank you for watching. Thank you for watching Biochemistry Unfolded, from atoms to algorithms. If you enjoyed this content, please subscribe to this channel, and we will keep you informed of future episodes. Thank you.